Welcome everyone to the ARM meeting on the Mesa. This is a virtual session to speak about the Bespoke Gene Therapy Consortium. I'm joined today by Courtney Silverthorne, who's the lead for the Foundation for the NIH in the Program Management Office, Peter Marks from the FDA, PJ Brooks from NIH NCATS, and Greg LaRosa representing Pfizer. The goal of today's session is to give you an update versus the session that we did at the 2020 meeting on the Mesa, where we introduced the Bespoke Gene Therapy Consortium to the ARM community. Since that time, just a year ago, we've made substantial progress in finalizing now this Bespoke Gene Therapy Consortium. And our goal today is to give you an overview of that consortium and also to answer some questions that you, the ARM community, may likely have about the consortium and how it will progress going forward. So with that, I'll turn it over to each of our panelists, starting with Courtney to introduce ourselves, then PJ, then Greg, and then Peter. Courtney?
Great, thanks so much, Bob, and thank you for the invitation to be here today. So my name is Courtney Silverthorne. I joined the Foundation for the National Institutes of Health about 4 months ago as an Associate Vice President in our Research Partnerships Office. And I am leading over the launch and execution of the Bespoke Gene Therapy Consortium. Before I came to the Foundation, I was at the National Institute of Standards and Technology. My career background is in technology transfer and intellectual property policy, and I also spent a number of years in research partnerships and technology transfer at the National Cancer Institute and at the Frederick National Laboratory for Cancer Research. So I'm very excited to be back in a biomedical space and looking forward to the launch of this important initiative.
Great, thanks, Courtney. PJ?
Yes, hi, my name is PJ Brooks. I'm the Deputy Director of the Office of Rare Diseases Research at NCATS within the NIH. And I've been leading the effort in starting up the Bespoke Gene Therapy Consortium from the very beginning and representing the NIH on it. And certainly looking forward to working with everybody and seeing the launch coming up hopefully shortly.
Great, thanks, PJ. Peter?
Hey, so I'm Peter Marks. I'm Director of Center for Biologics Evaluation and Research at FDA. I'm a hematologist oncologist by training, spent some time in industry, helping to run the clinical service of a cancer center, and then came to FDA. And we have been very happily partnering with PJ and colleagues at FNIH towards making the Bespoke Gene Therapy Consortium a reality, hopefully in the not too distant future.
Great, thanks, Peter, appreciate it. And Greg?
Hi, Greg LaRosa. I'm the Vice President of Scientific Research at Pfizer's Rare Disease Research Institute. And I've been there for about 10 years. And prior to that, a number of biotech companies in the Cambridge, Massachusetts area. My background is molecular pharmacology and molecular biology. And I've been involved with this also from the beginning of the conception. And I'm very excited that we're at this point now where we're ready to launch.
Great, thanks, Greg. And I'm Bob Smith. I'm responsible for leading Pfizer's global gene therapy business. And to round out our introductions, I'll turn it over to Courtney, who's going to give us an overview presentation on the Bespoke Gene Therapy Consortium.
Great, thank you. So wanted to give you some high-level overview of the consortium goals and future activities. And noting that the Bespoke Gene Therapy Consortium is part of our Accelerating Medicines Partnership Program that is overseen by the FNIH. And so to give you a little bit of broader context about the Accelerating Medicines Program, this is a series of public-private partnerships that launched in 2014. And the original 3 AMP programs were all focused on individual diseases or disorders. So our first 3 projects in AMP Alzheimer's disease, in type 2 diabetes, and in RA and lupus were all started in 2014 as a way to bring the public and the private sectors together to solve problems in developing therapeutics for various disease areas. All 3 of those original AMP efforts have since rolled over and or expanded into AMP 2.0s, so Alzheimer's disease continuing on in target discovery and preclinical validation. our type 2 diabetes program expanding out into common metabolic disorders, and our RA and lupus program into additional autoimmune disorders. Along the way, the foundation also started AMP partnerships in Parkinson's disease and schizophrenia. And the soon-to-be launched AMP Bespoke Gene Therapy Consortium will be our 6th Accelerating Medicines partnership. And we have a 7th that'll be coming online in early 2022. Across these projects, this represents over $670 million in financial and in-kind contributions from both the NIH institutes and our private sector partners. Across the programs, we currently have 13 NIH institutes and centers involved in various projects, 24 different industry partners, and 20 nonprofit organizations. So the Bespoke Gene Therapy Consortium is focused on 2 main tracks. The first focusing on AAV basic biology. And AAV was selected as the focus by the design team for the BGTC because it offered the most opportunities to evaluate and develop in the pre-competitive space. And so the design team identified a number of steps in both the vector generation processes for AAV, as well as the therapeutic gene expression processes in delivering these viral vectors to patients that they felt could be researched further, individually optimized, and potentially across the entire pathway of vector generation and gene expression, increasing the efficacy of these therapeutics potentially by orders of magnitude. The second component of the BGTC will focus on a clinical program, identifying various gene therapy targets that would be good opportunities to take AAV therapeutics into first-in-human clinical trials. And along the way, through the vector generation and manufacturing processes, beginning to standardize some of the steps in the vector generation, potentially developing some instructions for use for individual AAV vectors and different gene types, developing non-proprietary tools that enable the vector generation process. Again, in manufacturing, looking at leveraging existing expertise in manufacturing processes and protocols to start to harmonize the best practices for manufacturing AAV therapies, looking at developing standard process menus and known safety databases, and other information that would help facilitate the preclinical testing that's necessary for these AAV clinical vectors. In delivering these through pilot clinical trials, we hope to generate data that would allow streamlining of various regulatory pathways. And so again, looking for standard clinical and delivery protocols that could be developed for different AAV vectors, potentially even establishing drug master files with the FDA that would allow information to be used as reference material for future clinical trials. And both the AAV biology and the clinical programs have the goal of standardizing the process for developing these gene therapies for rare diseases, delivering things to patients faster and potentially at a reduced cost that would make some of these bespoke gene therapies more accessible. And so to provide a little bit more information about the AAV biology work that's anticipated under the consortium, again, the design team identified a number of different steps in the vector generation and the gene expression pathways that offered opportunities for a greater understanding of both the viral life cycle and how the genes are expressed in a patient. And so in the vector generation processes, the design team felt that different steps in the viral genome replication and processing could be researched and understood better, learning more about the capsid production and assembly processes, improving the packaging of viral genomes to generate more productive viruses and reduce the number of empty capsids, looking at different steps in the transport and release of the virus, and any host factors that might influence the process of the viral vector generation. And similarly, in looking at the therapeutic gene expression, taking a look at the endosomal state of the AAV virion and how it's trafficked and uncoded in the nucleus, conducting more research on the second strand synthesis steps, and looking at the concatemerization of the viral genome, as well as any post-expression events that might influence the level of gene expression in a patient. And so one can imagine that these could be done by various high throughput screens, various knockdown technologies, but really using these different steps as an opportunity to start to help overcome some of the existing limitations of AAV-based vectors and improving the knowledge of the interactions within the host at the cellular level, allowing us to improve the research body of work and improve more efficient transduction. And so as part of the clinical program, we'll be using a disease selection process for pilot clinical trials that we hope would provide data that allows us to streamline the overall process. And so the design team wanted to start this process from a disease agnostic state, and we'll actually be putting out a call to the academic, the research, clinical, and patient advocacy communities to actually submit or nominate studies on diseases or disorders to be selected for the BGTC clinical program. Out of the thousands of rare diseases, we hope to select 5 to 6 that will have a really high possibility of giving us good data at the end of the clinical trial that could be used for streamlining regulatory processes. At an individual level, some of the characteristics that the design team felt would be appropriate to consider. Obviously, we would want to identify diseases or disorders that have a clear monogenetic cause and would be amenable to AAV therapy. As all of our accelerating medicines partnerships focus on pre-competitive spaces, we're looking for opportunities that don't currently have a commercial business case. We'd like to see that there's sufficient existing information to run a successful clinical trial, in vivo data or patient natural history observations. Would be favorable to select things that have low trial requirements for testing and follow-up. So for example, a disease or disorder that could show success either through clinical progression or through an established biomarker in a matter of months rather than years. And similarly, diseases or disorders that already have a currently assembled patient group would allow us to enroll patients in a clinical trial much more rapidly. Also want to consider diversity and inclusion in selecting the diseases, ensuring that we have a diverse patient population. But we'd also like to look holistically across the clinical program and see diversity not just in the individual diseases, but also in the totality of the diseases are selected. And we'd like to leverage the existing expertise and capacity in manufacturing gene therapy product for these pilot clinical trials. And so what we hope to do here is to identify several vector manufacturers, some of which could be contributed as an in-kind contribution from some of our industry partners or contracted out for manufacturing. Potentially looking at different AAV serotypes and manufacturing scales, varying the routes of administration, something that's local delivery versus systemic delivery, and also looking across a number of different target tissues and diseases. But the goal of the BGTC is not to just do great research in AAV biology and improve the understanding in the field, not just to have 50 or 60 patients participate in a clinical trial, not even to focus just on developing 1 or 2 therapeutics through the FDA process, but really to use all of that information that's generated by the consortium to improve the clinical and regulatory frameworks for all gene therapies for bespoke disorders. So we know that currently most of the gene therapies that are developed for rare disorders are produced as one-offs. The manufacturing is done in a silo, the clinical trial is developed in a silo, and the regulatory submissions are submitted as a one-off function. And so there's no learning happening amongst all of the gene therapies that have been developed so far. And so what we need now is a more transformative approach to take the information that comes out of the consortium and use that to start to develop platforms and processes that will leverage this information, developing products, improving the knowledge for all of the bespoke therapies that come behind the initial clinical trials in the BGTC. And so what we hope is that we can use this information to standardize the processes, get things to patients faster, get things to patients in a reduced cost. possibly using these initial vectors as standards that could enable use of those vectors in a more rapid fashion in future clinical trials. And so one of the things that the consortium hopes to develop over the course of the 5-year program is a type of manual for AAV gene therapy clinical development compared to the Maniatis for cell biology. Really combining all of the learnings that come out of our AAV biology program, that come out of our clinical program, that we learn along the way in terms of streamlining the manufacturing processes for AAV therapies, and any regulatory improvements or streamlining that we start to develop. Bringing all these insights and learnings together to help facilitate the success of not just these 5 or 6 clinical trials, but all future clinical trials in gene therapy. And so some of the things that could be contained within this gene therapy manual, looking at different CMC testing requirements and optimized lot release methods and assays, and starting to harmonize the set of vector quality tests that are needed to move forward. Looking at the methods and criteria for how you select these diseases in bespoke and rare disease clinical trials, and compiling information about what a standardized regulatory submission package might look like. We also hope to have an extensive communications plan and have laid out 5 years of bringing BGTC information to communities like ARM and others. And so in the first year of the program, we'll primarily be talking about the launch and hopefully in very short order, various social engagement, media and launch materials, videos talking about the consortium, and continuing that into year 2, building out a website that contains explainer videos and infographics, and starting to hold annual meetings to bring together both the BGTC participants as well as others working in gene therapy and rare disease spaces. In year 3, we hope to be talking about some of the initial publications coming out of our AAV biology research. And in year 4, focusing on promoting the manual to the clinical and patient communities. By year 5, we hope to be talking about the impact that the BGTC has had on the entire field of bespoke gene therapies. And so one of the incredible things about the AMP BGTC program is that we have had phenomenal and widespread interest on both the public and private sector sides. So we're speaking and finalizing a number of potential partners, our full partner list to be announced at the launch, but we're proud to say that we have received sufficient commitments to meet our de minimis budget of $58 million, and we are rapidly moving towards a proposed launch sometime in October of this year. Looking to promote the opportunities to apply for the AAV Biology Awards, establishing various cores that will focus on CMC testing, the clinical trials and analytics, and beginning to look at the first 3 pilot programs, hopefully some that might be held at the NIH Clinical Center as well as at external sites. We're moving rapidly towards the full program proposal, which would allow for that full clinical trial program as well. And so this just gives you an overview of the expected timeline for the Bespoke Gene Therapy Consortium. Again, looking to launch within the next few weeks and releasing requests for applications for our AAV Biology Awards towards the end of this year. Also putting out information about the disease selection process that we hope to have the community participate in. In early 2022, we'll be making those selections for the AAV Biology Awards, as well as starting to finalize our disease selections for the clinical program and beginning to secure our vector manufacturing for the first 3 clinical trials pilots. Hoping to enroll patients in those first 3 clinical trials towards the end of next year, 2022. And looking at a go, no go milestone towards the end of the 3rd year of the consortium. So the steering committee will be looking at the results of the first 3 clinical trials, as well as the information that comes out of the vector generation and delivery research, making a decision as to whether the results are positive enough to hopefully go forward into years 4 and 5 of the consortium, continuing our vector manufacturing and clinical trials for our diseases or disorders, as well as starting to draft that regulatory framework and a manual to share with the clinical and patient communities. We expect to have some sub-teams established along the way that would support the consortium efforts in CMC testing and analytics, and in regulatory streamlining, as well as the communications and patient education program that will be putting out information through web and other media. So I'll pause there and turn it back over to Bob to go through our Q&A session. Thank you.
Great. Thank you, Courtney, for that excellent presentation. I'm sure many of the members of the OM community will have learned a lot from your overview. And again, congrats to you and the team at the Foundation for the NIH for getting very, very close to the finish line here with implementation of the consortium. Maybe the first question as we progress to answer a few, I think, important aspects of the Bespoke Gene Therapy Consortium. I guess, PJ, I'll start with you. I mean, it was really great to see so many of the different institutes within the umbrella of the NIH being involved in the consortium. Could you just give us a little bit of a sense as to how these different institutes are going to be involved in the participation of the consortium over the next 5 years?
Yes, thanks, Bob. Yes, we were very happy with the level of participation we got from many of the different NIH institutes and centers. I think it's a testament to their understanding and appreciation of the potential value of this program to gene therapy efforts as a whole that will undoubtedly then benefit their own programs. They all will be contributing funds which will go to various aspects of running the program. They will all be participating in the oversight and running of the program as part of the oversight committee. I also would anticipate that to the extent that they have up and running ongoing clinical trial programs, that we may be able to leverage some of those activities through these ongoing clinical programs that they already have, and that would certainly be a benefit to getting these trials up and running. And of course, some of them, as we discussed, some of these trials may be done at the NIH Clinical Center, that in a sense, all of the NIH institutes kind of already supply the funding for. So in that case, it might be some of their clinical investigators at the clinical center leading some of these trials. So we anticipate a variety of ways in which they will be involved.
Well, that's really great. I mean, and it's really interesting to see not just a broad interest, but the ways that they're going to contribute across multiple dimensions, including utilization of the NIH Clinical Center. So really glad to see such strong endorsement of the consortium from the member institutes and centers at the NIH. Greg, maybe a question for you. I know that you and Sang, representing Pfizer, have been sponsors of the overall consortium since the initiation of the concept. Maybe just share a little bit of your insights representing the broader industry participants in the consortium. You know, what's like driving people's interest to be members of the consortium, and what do you think some of the critical decision-making factors were for people getting interested in providing funding to support the consortium?
Yeah, sure. Thanks, Bob. I think, you know, there was really a lot of various reasons. I think, you know, most of the companies probably saw as a you know, value in this being part of this consortium. I think first of all, it's just, you know, you can work together with other, in a sense, with other companies and the NIH and the you know, the other institutes that PJ was just talking about. And rather than, you know, things being done in the AAV gene therapy space separately, and a lot of things being repeated, multiple different institutes, to be able to work together and try to solve some of the problems that we're all dealing with regards to AAV biology and manufacturing and running into, you know, getting into the clinic as well. So I think that's, you know, that's probably one of the primary reasons, is just being able to put some funding into this, but have it be matched really with a lot of funding from other sources to drive this forward, to help us answer questions that are still remaining. And there are quite a few, I think, in this field still. Another reason I think is that we're very interested in being able to not just learn more about the biology and the manufacturing and trying to streamline things and standardize things, all things which are needed and important, but also to be able to then utilize that to benefit patients that are suffering with rare diseases that are, as Courtney mentioned, that are really, they're just not commercially viable for pretty much any private for-profit organization, and these diseases have been relatively neglected. So this is a chance to sort of participate in being able to try to do something for those patients, especially in something that could be as transformative as gene therapy. So I'd have to say there's a number of reasons. Those are a couple that come to top of the mind.
Well, that's great. Thanks for sharing that, Greg. And I know that with respect to looking at the impact of this potential program to benefit patients with ultra rare disease, Peter, I know you've been a very enthusiastic leader of the consortium since its initial conception, and the importance of being able to make gene therapies available to patients in these underserved ultra rare diseases. Maybe you can just elaborate a little bit more on your perspective of your support for the concept of the consortium, and also maybe a more technically related question of how do you envision this concept of a originator product being able to be, quote, the maternal source of offshoot products, and how the streamlined regulatory framework that you're envisioning the consortium would implement could not only benefit the programs within the consortium, but maybe even beyond in the future, beyond the scope of the consortium.
Yeah, thanks very much, Bob. It's, the idea here is to try to leverage what we can in gene therapies that make them unique among other biologic products. And one of the things that's unique about them is that, at least for some classes, right, you're going to have the same backbone vector that's going to be used kind of as a workhorse. Now, it may be that that backbone will carry different payloads, different size payloads for different things, but it would be nice if we could find a given AAV vector that might have inserts of, accept inserts of a certain size. If we could leverage what we have in terms of knowledge of the vector and be able to use that for regulatory purposes to streamline the development process. And what I mean by that is, if we have a vector which we understand its toxicology, we understand how to go about manufacturing it at some scale, and how to purify it, how to characterize it. We'd like to be able to, instead of every time you put a new insert, perhaps of a similar size, into that same vector, same promoter, everything's the same except for the insert, that you could potentially be able to leverage all of this other information that you're otherwise repeating every time. The repeating of getting that information adds on a lot of cost to the process, plus it takes up time. And so the idea here is we could get to more diseases more quickly if we had higher throughput by leveraging something that's inherent in this class of products. So that's what gets me interested in this as a regulator. Whether it'll work perfectly, we don't know yet, but I think the hope is we can try this and see if we can make this work, if we can take advantage of this kind of almost unique aspect of gene therapies, and that they do have a structural element that is similar between different products that we can then, or can be similar between different products that we can take forward. It's true, you could say the same perhaps about some vaccines, but let's focus here on the gene therapy aspect of it.
Great, thanks, Peter. Courtney, maybe a question for you, since PJ had mentioned the oversight committee. If you could just take a few minutes to describe the governance and operational structure that you're envisioning the foundation for the NIH will operate under in terms of the execution of the consortium?
Sure. So while the Bespoke Gene Therapy Consortium is going to be the largest accelerating medicines partnership that the foundation has managed, the governance structure will be substantially similar to all of our other accelerating medicines partnerships. And so our steering committee for the BGTC will have a voting and a proxy member from each of our contributing organizations. All of the NIH institutes and centers, all of our private sector companies, and our nonprofit organizations will have a seat at the table. And all of our AMP programs have the voting that's distributed 50% to the NIH institutes and 50% to the private sector. And so that will be the case with the BGTC as well. We anticipate that there will be a number of subgroups and working groups that will be established by the steering committee for individual actions, carrying out some of the day-to-day activities of the consortium. And so the terminology that I'm using here, we describe a subgroup as something that's established for the duration of the consortium. And so I mentioned a few of those on the timeline slide that the design committee thought would be established pretty early up front, related to the CMC testing and manufacturing, related to the clinical trials oversight, the communications, and the regulatory streamlining. Other subgroups might be established by the steering committee as well. We also anticipate that there will be some working groups that will be spun up for a specific purpose and then dissolved once that purpose is completed. So initially, we know that there will be a working group that will review and make selections for our AAV biology awards. There will also be a working group that oversees the review and selection of the diseases for the clinical trials program. And again, additional working groups might be formed down the road, potentially one focused on drafting the AAV manual or other regulatory aspects. All of our accelerating medicines partnerships, the steering committees report up overall progress, updates, milestones to an AMP executive committee and an AMP extended executive committee that's made up of all of the co-chairs of our AMP programs, as well on the extended side, industry representatives as well. And so that happens on about a quarterly basis and will be in the regular reporting cadence of all of our AMP programs going forward.
Wow. Well, it sounds, Courtney, like you and your team have this in a very well-organized fashion, and it sounds like you'll be able to run it really efficiently. PJ, maybe one question for you. I know that you and the NCATS team at the NIH have spent a lot of time on the platform vector gene therapies project, including having some of that work published last year. Could you just give us some insights as to how using that PAVE GTX project will be able to maybe add some value to the consortium going forward?
Yeah, we certainly hope that it will. PAVE GT is designed to be entirely in the public domain, so we certainly would see a lot of opportunities for interaction and data sharing, and my involvement in both will facilitate that. I think a lot of the goals are similar, which is a streamlined regulatory pathway, though we can't accomplish that by ourselves with PAVE GT. We can kind of test certain possibilities. I think other things are a standard toxicology package for testing a new vector, a standardized set of assays for characterizing vectors. I think these are both things that are our goals for both PAVE GT and the BGTC. And quite frankly, we would like to see those efforts kind of support each other as much as possible. I think that would enhance the impact of both projects.
Great, thank you. And Greg, maybe a related question about not just what the NIH can bring to the table, what their various areas of expertise, including the PAVE GTX project work that PJ just described, but from a member of the private part of the consortium, where do you see industry participants and maybe even some of the academic groups being able to add value in terms of manufacturing of AAVs? It seems an area where industry's made significant investments, and how can we best leverage some of the benefits of those investments to the benefit of the consortium.
Yeah, well, I think that, it's meant to be pre-competitive. So certainly there's a lot of know-how that's been developed at the companies, but we want to find ways to participate in this whole, again, the understanding of the viral biology, the understanding of some, especially early steps in the manufacturing of growing the virus and maybe, increasing yields and so forth like that. Whereas maybe some of the downstream aspects of manufacturing might be a little bit more, a little less pre-competitive perhaps, but part of it can also be actually doing some manufacturing for this, as Courtney mentioned, that as in-kind contributions, some of the partners could actually do some of the manufacturing for those trials for that Courtney again mentioned in the ultra rare. Some ultra rare indications that the consortium will choose. So I think it's trying to bring what we've learned all from all these different industry participants, as you said, in the huge investment that's been made, I think, in a lot of institutes for manufacturing AAV, and to try to bring those together to help move this forward and help our understanding of how we can, again, standardize things and increase yields and hopefully decrease the doses as well.
Great, thanks, Greg. Courtney, I don't know, I see you're nodding your head there. Did you have something that you wanted to add to Greg's response there?
No, I just think it's such a great opportunity for the incredible manufacturing expertise and capacity that already exists at some of these companies to be leveraged to move these clinical trials forward. And we hope to engage other organizations that focus on manufacturing and potentially have additional information that gets put in the public domain to advance these therapies.
Thanks, Courtney. Peter, a question for you on the envisioned Bespoke Gene Therapy Consortium Clinical Development Manual. Could you just describe a little bit for the audience how you envision that being potentially different than the existing set of FDA guidance documents that exist for gene therapies? Is this something that is going to be complementary, or just give us your perspective on how you envision this being used not only in the consortium, but maybe even beyond outside the scope of the consortium?
Yeah, so I think this is one of these things that it doesn't have form on it yet. So take everything I say with a grain of salt. But there's what we have in guidance, which says what it's supposed to look like. But I think the real issue here is there are a lot of academic investigators out there that do things differently. It would be nice to have a manual that helps people understand the most efficient ways to do the clinical development programs for these, with a variety of perhaps different options depending on the nature of the effect that's being observed, whether there are any surrogate endpoints or intermediate endpoints that might be applicable that would allow one to leverage an accelerated approval pathway. So trying to make this easier so that people are not trailblazing every time. Yeah. I would think also that this would help the academic or even the small company understand what is necessary in terms of enabling material to get towards clinical trials as one submits an investigation or a drug application and then take it through the process. So I think the difference here is the amount of granularity that would be present.
Great, thanks, Peter. A very noteworthy goal. So hopefully this will really be something that'll benefit way beyond the scope of the consortium. Courtney, we're winding down to our last minute or two here for our allotted time. Maybe I'll turn it to you as a program manager for the consortium to share with the ARM member community if they're interested in getting engaged with the consortium, how best can they do that? I know that a lot of members have already signed up, but we're more than welcome. We're more than willing to welcome more members, but I leave it to you to just describe to the audience, how best they can get engaged with you and the consortium.
Sure, thanks, Bob. So there are a couple of different ways that we see engagement with the consortium. One obviously is to become a consortium participant, and even past the launch of the consortium, we expect that there will be continued interest from potential future partners, and we're happy to continue the conversations on what that participation might look like with other organizations. So if any of our members are interested in finding out more about that, on the FNIH website, fnih.org/BGTC. They can see an overview of the program and also my contact information if they'd like to get more information about being a contributing member to the consortium. There will also be other opportunities for external parties to play a role in the activities of the consortium. Obviously, recipients of the AAV Biology Awards will play a very big role in carrying out the important research on the vector side. The communities who submit diseases and disorders to be considered for inclusion in the clinical program will be very important, and we encourage our membership to look at both of those opportunities when they come out following the launch. There will be opportunities along the way for external subject matter experts to be included in some of the sub-teams and working groups. And so as the steering committee determines those opportunities, they'll be inviting potential members to join. And then through our meeting and advocacy programs, we'd like to have very widespread participation, joining us at the table to learn from the BJTC's efforts, as well as to bring some of the learnings of other communities into the consortium as well. And so we encourage people to look out for those opportunities as they start to come.
Great, thanks, Courtney. And I'd just like to take the last minute here to thank ARM for the opportunity to convene this panel, to share more about the Bespoke Gene Therapy Consortium, and most noteworthy to thank Courtney, Greg, PJ, and Peter for their participation, sharing their insights and thoughtful responses to our questions. So thank you all very much and enjoy the rest of the conference. Bye-bye.